In this video, we're going to be talking about the biochemistry of carbohydrates. So a lot of the material that we've learned in this lesson, we've already learned in our biology course, but we're going to go into some of the finer details surrounding the stereochemistry of carbohydrates. First of all, we'll talk about monosaccharides. So monosaccharides are simple sugars, and they can range from 3 to 6 carbon atoms long. And they have the general formula CXH2OY. And in monosaccharides, the X and Y are the same. So if it's C3, it'd be C3H6O3 or C6H12O6. That formula will stay the same for disaccharides. However, with each combination of two monosaccharides, you lose a water. So the X and the Y will be different for disaccharides. But we'll talk about that later. Every single carbon has a hydroxyl group, except for one. And that one carbon has a carbonyl or carbonyl atom instead. So remember from biology, we have aldoses. Aldoses have the carbonyl on the first carbon, making them an aldehyde. And then ketoses have the carbonyl on the second carbon, making them ketones. All monosaccharides are reducing sugars, which means that they can be reducing agents because of that carbonyl functional group. And we will learn a little bit later in the lesson how we can test to see if a sugar or a carbohydrate is a reducing sugar. There's actually a test that you can do in the lab. So on this diagram, we have two different hexoses, so 6 carbons long. The first one is an aldohexose because of the carbonyl being on the first carbon. So that's actually glucose. And you can see that when you look down the length of the molecule, the hydroxyls are all on the right except for one. And then this over here is fructose. And you can see here the carbonyl is not on the last carbon or the top carbon in this molecule. Instead, it's on the second one. So this is a ketohexose. So fructose is on the right here, and glucose is on the left. When carbohydrates and monosaccharides are in their dry state, they're linear structures, as you can see here on the left. However, when they're in solution, they fold into a cyclic shape. So what happens is this hydrogen comes up, breaks the bond, and then we get this ring structure. Their molecular formula is the same, but now they're isomers of themselves. So even though they're both glucose, one is in linear and one is in ring structure. The cyclic or ring structure is a more stable molecule. Now, you don't have to memorize these molecules at all. They're all listed in section 34 of the data booklet, both the ring and the cyclic structures, or both the linear and the cyclic structures. There are 3 different ways to represent monosaccharides. You can represent them in their straight chain form. So this is fructose in the straight chain. That is called the Fischer projection. When they fold into their cyclic structure, that is called the Haworth or Haworth projection. However, neither of these actually show the bond angles. They're easier to draw and they're easier to look at, but the final one, which is on our next slide, is the chair and boat drawings. These actually show a little bit more accurately the bond angles involved. They're more accurate, but they're not as easy to draw, so we won't be using these structures in our drawings and discussions. So the properties of monosaccharides. Because of all these hydroxyl groups here and everywhere, that makes them polar. So that means that they're soluble in water because they hydrogen bond with water. So there's one hydrogen bonding here. There's another water molecule hydrogen bonding there, and so on. So because they're soluble in water and because they're polar, they're easily transported in blood. And that's a really good thing because we need to be able to transport our nutrients throughout our body via the bloodstream, so they can be dropped off to the tissues that need some carbohydrates. And then we can also use carbohydrates in pharmaceuticals because they can bind to medications that are usually in crystalline form, and then they can be dissolved in water and then transported to the cells and tissues that need the medication. And then the final one is the one that we're most familiar with. They are used to sweeten food. We like to sprinkle carbohydrates on, our breakfast cereal, in our coffee, in our tea, and so on. In biology, you may have remembered that one of the skills that you need to do is to draw alpha D glucose. You probably wondered what the alpha and the D are, so we're going to be talking about that for the next little bit. Sugars, because they have more than one chiral carbon, means that they are optically active. So anything that's considered an L sugar will rotate plane polarized light to the left, so levorotary. And then anything that's a D sugar rotates plane polarized light to the right, dextrorotary. So D for right, L for left. So how do we know if something is a L and a D sugar? Where do you look to find out if it's one or the other? So this concept was first noted in glyceraldehyde, a 3-carbon sugar, by Emil Fischer, which is where we get the Fischer projection from. So if you see here, this is a pretty simple sugar. It's only 3 carbons long. So there's our central carbon. And you can see that it's chiral because there's 4 different things bonded. And you can see that this stays the same and this stays the same, but the position of the hydroxyl and the hydrogen alters, and that's what makes them D and L. So we're going to go into a little bit more detail on our next slide. So if you needed to identify whether a sugar is a D or an L projection, you look at the furthest chiral carbon from the top. So the top we always have wherever the carbonyl is. So this is the top of the molecule. You can see every single carbon here is chiral, but this is the one we're going to look at. So this chiral carbon, if the hydroxyl is on the right, it is the D configuration. So this is D glyceraldehyde. If the hydroxyl is on the left, then it's L glyceraldehyde, or L-glucose. So you can see there's 2 different sugars being used here. So this is the bottom one. Remember, glyceraldehyde only has 1 chiral carbon. They're both on the right, so they're the D configuration. So D-glucose, D-glyceraldehyde. And this is L-glucose and L-glyceraldehyde because that last chiral carbon has the hydroxyls on the left. So the question is, where does alpha and beta come from? Well, when a monosaccharide is in its linear form, the carbonyl is not a chiral carbon because it has a double bonded O, so it's not bonded to 4 different things, it's only bonded to 3 different things. But remember, when the sugar folds into the cyclic form, the carbonyl disappears, and then we now have a chiral carbon. So this is chiral now, even though it wasn't before, because there's a H and OH, all of this bonded and all of this bonded. So it is chiral. And there's 2 options for the arrangement. It could fold and have the hydrogen at the top and the hydroxyl at the bottom. Or, as on the right here, the hydroxyl went to the top and the hydrogen went to the bottom. So if the hydroxyl points up, like this one, it's the beta form. And if it points down, it's the alpha form. So remember, these are all in your IB data booklet in section 34. But in biology, you have to be able to draw this structure right here, the alpha D glucose. And remember that most sugars form equal amounts of both of these anomers, which is just another word for isomer. Again, in biology, we learned about disaccharides. So this is all pretty much the same as what we learned before. So 2 monosaccharides are formed or joined together by that process of condensation reactions. Remember, the way I remember is we're condensing 2 into 1, and it's forming some condensation. We're producing water as a byproduct. So what happens is a hydrogen and a hydroxyl interact, forming a bond, and then you end up with a disaccharide plus one water molecule. The bonds are called glycosidic linkages, and the general formula is still CXH2OY, but because, like I said before, we lost one water molecule, the value of Y is one less than X. So it would be, like if it's two glucose being joined together, or two other hexoses, it would be C12H22O11. So the Y is one less than X. So again, we learned all of this information in biology as well. So maltose is two glucoses joined together. We find them in beer and malted barley products. So we don't tend to use those for sweetening. Lactose is a combination of glucose and galactose, and we find those in milk products. We're going to find out a little bit more about lactose and lactose intolerance a little later in the lesson. Sucrose is glucose and fructose joined together, and that's found in sugar cane and sugar beet. So that's the sugar that we sprinkle on our food. So it's quite sweet. The other two aren't overly sweet. We are conditioned to taste the sweetness of sucrose, and we are programmed also to like that flavor. So humans tend to really like to taste sweetened things. So lactose intolerant. Normally, mammals stop drinking milk after infancy when they get weaned off of their mother's milk. So then that stands to say that they generally stop producing lactase as well, because you don't have to digest lactose anymore. But as humans, we really like milk, so we continue to eat products with it. But if we're not making as much lactase, that means that we can become lactose intolerant. And that is actually genetically linked. And you can see that there's distributions throughout the world where there's higher instances of lactose intolerance. But because we like milk so much and we don't want to give it up, we found ways to reduce that lactose intolerance. We can take enzymes to help us digest the lactose. We can reduce the lactose in dairy products, and so on. So the properties of disaccharides. Again, because they have all those hydroxyls, that makes them polar. So remember, the functional group in carbohydrates is the hydroxyl. Again, in their solid form, in their dry form, they're crystalline solids. But because of the hydroxyl groups and because they're polar, they're very soluble in water. And we all know that because when we stir our sugar into our tea, it disappears and spreads throughout the solution. Now, disaccharides that were made from aldoses still remain reducing sugars, because aldoses can go back to their straight chain form, but ketoses can't. So that means that lactose and maltose are reducing sugars because they're made of either two glucose or a glucose and a galactose. Those are both aldoses. However, sucrose is not a reducing sugar because fructose is there. So fructose, remember, is a ketose. So now it can't go back to its linear straight chain form, so it cannot act as a reducing sugar. So we can test for reducing sugars, and you can do this in the lab if you have some Benedict solution. So Benedict solution is a reagent that you can use to test for reducing sugars. So you put some of the Benedict solution in a test tube, and then you put a sample of what you're trying to determine whether it's a reducing sugar or not. And there's a redox reaction that happens. So there are copper 2+ ions in the Benedict solution because there's copper 2 sulfate as one of the main ingredients. And it will react with the aldehyde group in the sugar to form copper 1+ and a carboxylic acid. And it actually forms an orange precipitate, or a reddish orange precipitate. So your solution will turn from a transparent turquoise to an opaque orangey red. So then if that happens, then you know that there's reducing sugars in your sample. Again, polysaccharides should be familiar to you. Those are due to the fact that monosaccharides can actually form polymers. So there are two classifications. If the monosaccharide has polymerized into 3 to 10 sugar molecules, then it's determined to be an oligosaccharide. If it's longer than 10 monosaccharides, and that can go up to hundreds or even thousands of monosaccharides long, then it's considered a polysaccharide. There are 3 polysaccharides we'll discuss, actually kind of 4. So the first one is starch. That's the polysaccharide that's found in plants that's used as a storage molecule, so an energy storage. The starch in this case forms alpha-1,4 glycosidic linkages. So the linkages are happening between carbon 1 and carbon 4. So carbon 1 on this one and carbon 4 on that one. And they're alpha linkages, so that means that the hydroxyl is pointing down. Starch is found in 2 different forms. The first one is more soluble than the other. So this is amylose. So you can see it's just straight chained and it actually forms like a helical shape due to their properties. A myelopectin is more branched and it's less soluble, but both are still water soluble. But we actually use pectin on purpose to make jams and jellies. So we can actually test for starch. We use a solution called Lugol's iodine, and if you're familiar with iodine in its normal state, it's a browny orange color. And if you mix it with starch, which is in test tube sample B, the 2 mixed together, you actually end up with a kind of a blackish blue color in the presence of starch. So cellulose is a plant polysaccharide, again, but this time it's not for energy, it's for structure. So because most animals don't have enzymes to hydrolyze or break down cellulose, it acts as fiber in their diets, and that's actually an important component of our diet to help keep things passing through our digestive system. It actually helps absorb foods that contain cholesterol too, so there's some benefits of eating fiber even if it doesn't give us energy. Cellulose are long intertwined linear chains, and they have really strong intermolecular forces between them, making them insoluble in water. And as I mentioned before, cellulose makes up the cell wall. It's a structural component, not an energy component. Our last polysaccharide we'll discuss is glycogen, and it's very similar to starch in that it provides energy storage. However, it's produced by animals and is stored in the liver and the muscles of animal tissues. When an animal needs extra energy, it will hydrolyze the glycogen and the glucose is used. And if you remember from biology, glucagon, one of the pancreas's hormones, is involved in hydrolyzing and like mobilizing that glucose. But you don't need to know that for chemistry. And then glycogen is much more branched than starch, but it still is water soluble. So you can see the 3 differences: linear for cellulose, branch for starch, and then highly branched for glycogen.